Hello, and this is our lesson over dihybrids today. Before we start dihybrids, we will do a quick review over our sex-linked inheritance. This question is a matching question, and hemophilia is a sex-linked recessive trait. If you have the dominant big H, you do not have hemophilia, but if you have only little h alleles, you will have hemophilia. Remember that sex-linked traits are carried on the X chromosome. For this problem, you'll drag the choices down and choose where they go. And if you don't like where you placed it, you can always click the little X button there. We'll put these in their places. We'll check your answer. There's a multiple choice question. And one more multiple choice question for you. So you're trying to figure out what the red square is on this Punett square by distributing your alleles from the parents. And this one's asking you about the question mark here, multiple choice question. And it is also talking about hemophilia, so it is a sex-linked recessive trait. And this one's asking you if they're having a daughter, what's the chances of their daughter being colorblind? For a dihybrid trait, it is a two-trait cross. We're looking at two different characteristics at the same time. So far, we've been doing all the different monohybrid problems, single trait crosses. This time, we're doing two-trait crosses. First of all, probably one of the hardest things is determining the parental genotypes, figuring out what their genes are going to be. And then we're going to determine the gamete combination. So the parents can only pass you one of their two possible alleles or genes for that chromosome or that trait. And they have to segregate those alleles. For example, for the big R, big Y, little y, they'll only be able to give you one of the R's and one of the Y's. So when we determine the gametes, we're going to use a foiling technique to show all the possible combinations of R's and Y's we can have. And then we're going to set up our Punett square. It's a 4x4 Punett square, so 16 square Punett square. We're going to draw 3 lines down and 3 lines across and put all the possible gametes for the one parent on top and all the possible gametes for the other parent on the side and distribute, calculate. When you FOIL, you're going to take the first letter of each letter set, and then you're going to take the first of the first letter set and the last of the second letter set, and then you're going to take the second of the first letter set and the first of the last letter set, and then you're going to take the last of each letter set. So if we had big H, little h, big S, little s, the first would be big H, big S. The outside two, first and last, is big H, little s. The inside two would be little h, big S. And the last of the first parent's letters would be little h, little s. So you definitely have to FOIL for parents who are heterozygous. For parents who are homozygous recessive, like little h, little h, little s, little h, little s, little h, little h, little h, little s, little s, it will give you all the same combinations, a little h and a little s. So if they're homozygous dominant or homozygous recessive, technically you don't have to because it will give you the same combinations. Remember, we've talked about Gregor Mendel. Well, he's still in play here. He was using dihybrid crosses with his pea plants, and he needed to answer one more question. When alleles are being segregated during gamete formation, does the segregation of one pair allele have any effect on the segregation of a different pair of alleles? In other words, does the gene that determines if a pea plant is tall or dwarf have any effect on the seed color? And he designed a second set of experiments to follow two different genes as they pass from parent to offspring. And this is our two-factor cross or a dihybrid cross. And in his results, the answer is no, they do not influence each other. One parent had peas that were round and yellow, and the other parent had peas that were wrinkled and green. The round and yellow traits were dominant. First Mendel crossed the true breeding plants, brown and yellow, big R, big Y, big Y, with wrinkled green, little r, little y. And all his results were big R, big Y, little y. And that showed that all those offspring produced from a green pea crossed with a yellow pea were going to be, ended up being round and yellow. And he was like, where did the wrinkled and green trait go? So then he self-crossed or crossed the two hybrid from the Feli 1 generation or the first generation. So he crossed two heterozygous ones, big R, big Y, little y, had them self-pollinate there, and then he was able to see the distribution of those alleles, showing different combinations of round yellow, round green, and wrinkled green. So his work, remember, formed the basics of this genetic for us and showing that genes are passed from parents to offspring. Each gene has two or more forms called alleles. Some alleles are dominant while others are recessive. Each parent has two alleles for the particular trait. They inherit those from the parents. They only pass one. So they're segregated in the gamete formation of sperm and egg cells. The alleles for one trait segregate independently from the alleles for another. So it didn't matter if they were green, they weren't always going to be round. This is similar to our Punett cross. This would be like crossing two heterozygous parents. In this case, we're looking at cats. And so the first step would be to foil the parents' genes to get their gametes. At the top parent, if we foil, so these are big S, little s, big B, little b. We write that out. I like to write it in like parentheses, so I have big S, little s, big B, little b. The first of each parentheses is big S, big B. The outside two of each parentheses is big S, little b. The inside two is little s, big B. And the last is little s, little b. You take all that information for the one parent and put it up top. And then they did also cross another heterozygous parent, so that information will be the same. Goes on the side, and then you carry them down and across. What's nice about the dihybrid squares is we're back to like Mendel's complete dominant pattern. So anything with a big S is going to, in this case, be white. Anything that has all little s's would be brown. And in this situation, anything with a big B will have a long tail, and anything with little b's will have short tails. We have my letter switched. So anything with a big S is going to have short tails, and anything with little s's is going to have long tails. Anything with a big B will be brown. Anything with all little b's would be white. So 9 of them are going to be brown with short tails. 3 of them, and that means they're dominant for both traits. 3 of them will have the dominant for the first trait, but recessive for the second trait. So they're going to have short tails, but they're going to be white. And then 3 of them will be recessive for the first trait, but dominant for the second trait. So they're going to have long tails, but they're going to be brown. And then 1 of them will be recessive for both traits, having a long tail and being white. To remember the patterns, when we crossed like that 2 heterozygous ones in a monohybrid scenario, it was always a 1-to-2-to-1 ratio, 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. The patterns are true also with the dihybrids. If you cross 2 heterozygous, it's always going to be a 9-to-3-to-3-to-1 ratio. And this is back to Mendel's P's. So you can see those combinations here. If you're crossing that self-pollination of big R, big Y, little y, the first step would to be knowing that they're heterozygous for their traits. So we know this is the parent. And we foil to figure out the gametes that are on the side of the square. So we're trying to figure out what these are, if we didn't know already. The first of each parentheses. big R, big Y. The outside of each parentheses, big R, little y. The inside of each parentheses, little r, big Y. And the last of each parentheses, little r, little y. Goes on the side. He's also crossing this again with the other parent. That would go here. Then you carry them down and across. So how you calculate the results. The first thing you'd be looking at, like how many have at least a big R and a big Y, and this would be dominant for each trait. And that is here. Right. Then he would like know how many are dominant for the first trait, but recessive for the second trait. So at least a big R, but then all little Y's. VOM is dominant, REC is recessive. So we have here, here, and here. There's 3 of those. And then he'd like to know how many are recessive for the first trait, all little R's, but at least a dominant for second trait, at least a big Y, at least one big Y. So we have one here. We have one here, here, here. So there's 3 of those. And then we have this one. There's one that's recessive for everything. Pass out all lowercase letters. So remember, trying to figure out the gametes, you FOIL, and then you're going to arrange those at the top or the side of the Punnett square. And all of the parents' gametes are remote either at the top or either at the side. This is crossing black versus white fur, rough and smooth fur texture. They're crossing 2 heterozygous ones. Those parents show black rough fur. So if you FOIL all of like the dad's gametes are up top and all the moms are on the side or something like that, and then you distribute them. This is asking how many would be black and rough, black and smooth, white and rough, white and smooth. Black being dominant, so it'd be anything with a big B, anything with a big R. It could be big B, big R, big R, big B, little b, big R, little r, big B, big R, little r, big B, little b, big R, big R. Black and smooth would be anything with a big B, but all little R's. So big B, little R, or big B, big R, little R. And you have white and rough, all little B's. And then big R, or big R, little r. And then white and smooth is little B's and little R's. So you want to know how many are black and rough. Black and rough are the dominant characteristics, so it has to have at least a big B and at least a big R. Black and smooth, at least a big B, so big B, or big B, little b, but then all little R's. White and rough, so you have little b, and at least a big R. So little b, big R, or little b, big R, little r. And then how many are white and smooth? So little b, little r, little r. This shows you the results. So anytime you cross heterozygous ones, you're going to end up with this same scenario as 9-3, the 3-to-1 ratio. And then this has some practice problems figuring out like from the words, what will their gametes be? And this one says it's a purebred dominant one. So based on knowing that it's purebred dominant or homozygous dominant, what will both of their traits code the B? And they might ask you like, okay, what's going to be box 3 if you have this dihybrid square? So practicing distributing your alleles. So it's having you practice tallying, and you're going to pick how many of them is going to have little r, big B, or big B, little b. So when it says big B dash, it doesn't matter what the second one is, as long as it has at least one big B. So you'll drag your number to the box, or you can click it. And you'll be able to continue practicing those. There are some more questions that you can do after class with the work here. After class, there's a traditional dihybrid worksheet you can look on doing, and it has the boxes already made for you, just down below the question. And you can work on tallying that. There is a Amoeba Sisters video and worksheet that you can watch and do, or you can just watch the video. And there's also additional review for sexing traits and pedigree. And then the vocabulary, overall vocab, Quizlet there. And just some other quizzes games if you want to play, just with them, just only multiple choice. And then this is a interactive game you can play. It's a pigeon genetics activity. Yeah. So let me know if you have any questions. Feel free to practice the quizzes. That's my main suggestion. And then I would recommend working on one of the worksheets or the Amoeba Sisters video. There's 2 different worksheets. This one is just problems, and then the questions are to the side. Just has 4 problems. And then there's additional expansion problems if you felt like doing those. So feel free to message me, and if you have any questions along the way, let me know, and I'm happy to help you. If you want to turn in anything, feel free to let me know, and I will grade it and send you feedback. If you play a quizzes game, Quizizz, just send me your username. The Quizlets, I can't see your results, so make sure you take a screenshot or record your score. Let me know if you need a certificate or anything. Thank you for joining me. Bye.